Welcome to the final panel breakout here at Field Advisor Forum. I'm Stephanie Porter, and I'm the Outreach Agronomist with the Illinois Soybean Association. Thank you so much again for joining us today. Our discussion, obviously, as you can see on your agendas, is on soybean cyst nematode management. And I'm very excited to be joined by two panelists, Dr. Nathan Schroeder and Dr. Matthew Hudson. We were also supposed to be joined with Dr. Jason Bond. Today, he unfortunately has lost his mother, so he was unable to join us today, but we send our deepest sympathy to him. Some quick housekeeping notes. If you're a certified crop advisor, please sign the sheet available in the room to claim your CEU credits after the session. This session will be live streamed on Field Advisor YouTube channel, so feel free to share the link with anybody who might want to join us remotely. And now we'll just get to it. How about I introduce our panelists? Dr. Matthew Hudson is a professor of crop sciences at the University of Illinois with affiliations across multiple research centers, including the Center for Digital Agriculture, the National Center for Supercomputing Applications, and the Institute for Genomic Biology. His work focuses on applying digital and genomic technologies to crop science, with a special emphasis on discovering mechanisms for SCN resistance in soybean and strategies to address SCN virulence. Dr. Nathan Schroeder is an associate professor of nematology in the Department of Crop Sciences at the University of Illinois Urbana-Champaign. Over the past decade, Dr. Schroeder has been a key faculty member at UIUC, teaching plant pathology and conducting research on nematodes. His work includes both fundamental studies to better understand nematode biology and applied research aimed at developing effective control strategies for plant parasitic nematodes. Please join me in welcoming Dr. Hudson and Dr. Schroeder. So first off, I don't want everyone to assume, but we are funding, or I should say ISA is funding projects with both of these individuals. So I'm going to go ahead and let them share with you, just an overview of what kind of research that they're working on. And any kind of, if you want to go through your slides, that's fine too. We'll just go with the flow here. Okay. How about that?
Sounds good.
And the questions will kind of go, I think, after you guys share some of your information. And one thing I want to point out, because I think it's super cool, is, and I want you to elaborate on more, Matt, on this, but one of our board members actually initiated the project that you're working on.
Yeah.
Correct.
And
go ahead.
I'll talk a little bit more about that in my slides. Actually.
Okay, awesome. Thank you.
I'll let Nate go first, but I think it's really great to have this kind of engagement, and it's really it's surprising to me that actually the ideas coming out of the Illinois Soybean Association are more forward-looking than we're getting from the federal government and other places. So that's really exciting.
Okay, very good.
Yeah, so for the past year or so, over a year now, we've been conducting free testing for SCN for Illinois growers, so funded through ISA. The samples come in, they're processed by our plant clinic here at the University of Illinois for number of eggs per 100 cubic centimeters of soil, and that's sort of a standard metric. And so this first slide is showing the results there, the number of positive samples per county, so it's somewhat color-coded, red being sort of the highest average counts. Gray indicates we have no data from those counties, so if you're in one of those counties, please contribute. And I think the take-home message from this is one that probably many people are familiar with, is that SCN is here. We have it in Illinois. And then the other one is that it's very patchily distributed, which we see both at a field scale as well as a statewide scale, that we can't just predict, oh, SCN will be there and it won't be there. So we get very, very high numbers in some fields. And even for some farms, we had one farmer who submitted 4 samples. In 2 samples, we had zeros. In 1 sample, we had about 320 eggs. And in the 4th sample, we had over 30,000 eggs per 100 cubic centimeters. So huge variation in distribution. So lots of data suggesting SCN is here, it's problematic. We are doing actually a pretty good job. So the highest number we got was about 48,000 eggs per 100 cubic centimeters, but about 20% of our samples were zeros. So we are doing a good job controlling it. Matt is going to talk more about the mechanisms of resistance, but I did want to share one slide, which was a survey. So Matt will probably talk more. I was told to point this at the computer. There we go. So one of the main mechanisms for control or control strategies is plant disease resistance. And the issue that we face, and Eliana talked about it earlier in her talk, is that we've been using the same source of control for the past 30 years, which is a source of resistance called PI 88788. And I did a survey of about 15 companies, 650 varieties, from maturities group 2 to 4.9, and about 90% of them have that 88788 form of resistance. So there are Peking forms of resistance, which is a different form of resistance, and again, Matt will talk more about that. Surprisingly to me, there were some susceptible varieties out there still on the market, which is unexpected. And really, there's very few, only 3 varieties outside of 88788 or Peking that were resistant. And actually, I believe, Eliana, those came from the Baird seeds. And I believe they were non-GMO varieties. So, and then the other issue to note is if you're planting in southern Illinois, no Peking varieties for maturity groups 4 and up. So with that, I'm going to just pass the torch on to Matt, and he can talk about some of his work.
Thanks, Nate. So I was going to talk about a lot of these things. I'll touch on them, but I was asked to specifically talk about the Illinois Soybean Association project, which is new. So I'm going to talk about that a little bit, but I'll just introduce what I'm going to say. So I don't know if I
think you have to get closer.
Okay, yeah, I might need to stand up over here. There we go. That's not the first slide. Okay. So I want to talk about control strategies for cyst nematode. And as Nate mentioned, we've basically got one that really works, which is resistance genes in the plant, right? So that's been our mainstay to keep the nematodes down, that and rotation. So and we have good evidence of this from our North Central project that I think was what Nate was referring to. If you grow the same soybean varieties with the same resistance source year over year over year, you're going to see the nematodes adapt to that. And we are looking and figuring out why they adapt to it, why they adapt to some types of resistance more quickly than others. And we've had a good run with this PI 88788, but it seems to be coming to an end. So it's not working as well as it used to in Illinois. Most of the samples we get seem to be able to overcome that type of resistance now. We've got these other varieties, these Peking type varieties, but they're not as high yielding. They're usually not available in like the latest herbicide resistant technology and things like that. So there's reasons people don't want to grow that Peking type varieties or these other resistance type varieties. What other alternatives have we got? Well, there's nematicides. We've kind of been around that one a few times. The companies have tried that. They've also got a mixed record. The resistance in the plant tends to work better. They tend to be toxic. They're bad for the environment. Some of them work okay as a seed treatment, but mixed results with nematicides that target nematodes directly. We've got a USB project that looks into enhancing the plant's resistance chemically. So can we add chemicals to the mix as well as the genetics to make our resistance last longer, make it work better? But what the new ISA project is, which came out of farmer input, is, well, why don't we think outside the box a little bit? and look at some of these new technologies that have come along that might allow us to actually target the nematodes using biological methods rather than chemical methods. And the idea there is the original thought was there's this technology called a gene drive. And the idea behind a gene drive is we have this gene editing technology now, CRISPR, and the gene editing technology is itself a gene. So you have a gene that can hop out of the place that it's in effectively as an RNA, and it can go and edit other places in the genome. And it turns out it can also edit other genomes in a cross. So this has been mostly thought of as a way to target insect pests like flies and mosquitoes that cause disease. The idea is that you take a gene, you put it into some flies, usually male flies. They're going to go breed with a bunch of female flies or female mosquitoes. The offspring, that gene edit is going to activate, and it's going to copy itself into both genomes. So then all of the offspring from that cross will all have the gene drive. And then their offspring will all have the gene drive. So the gene drive duplicates itself through the whole population. It spreads very quickly. And it can then deliver either a gene that causes usually the flies to be infertile, so they then can't breed. So that'll knock down the population massively, possibly even causing that fly to become extinct is the idea. So if something like an Anopheles mosquito that carries malaria, people wouldn't be too upset if that got extinct, right? And the other possibility is you can put something in there that specifically targets it, makes it susceptible to some kind of other thing like a chemical intervention. The problem with this technology, there's a few issues with it, but the main one is you have to keep that gene drive down while you're actually breeding the flies before you can release them. So you somehow have to make lots of flies to release. You can't do that if they're infertile. And the other problem is that because it's designed to spread, it's designed to spread through the population, it's designed to edit genomes, people are a bit nervous about that, right? We don't know how far it's going to spread. Could it spread to other species? Could it spread to like birds that eat the mosquitoes? And maybe it could spread in a way that actually made the mosquitoes more resistant to pesticides because it's designed to mutate them. Maybe it could have an unintended consequence that actually was opposite to what we intend. So there's a lot of risks with it, and for that reason, it's not actually been approved for any kind of release anywhere in the world yet. There is another technology which this company from Britain, Oxitec, came up with, which is similar, but it doesn't have that spreading aspect. So what they do is, again, with mosquitoes is the original target for this, but they're targeting more insects, and it could work in nematodes. You have a protein that basically causes a gene that the mosquito really needs to produce, and it knocks that gene down so that they can't reproduce anymore. And it does that via another gene. It doesn't spread, but that gene goes in through the male line. So you have to breed a lot of male mosquitoes with this gene. And there is an antibiotic that you can give the flies that stops that gene being expressed. So you grow the flies on antibiotics, rear lots of them, release them, and then those males will mate with females. Their offspring won't be viable. So, and this actually has been shown to work really well, and it's actually getting quite widely used in Africa as a way of controlling mosquitoes. So there's some technical challenges with getting this to work in worms. One of them is our gene insertion, the transgenic technology where you can put transgenes into the nematodes is not very good. And doing that stably in male nematodes is not very easy. Also, you can rear these flies on bananas, right? You just pulp up some bananas, or the mosquitoes you have to use, I think they use pig blood or cow blood. We have to use soybean roots. So you have to grow an awful lot of soybean roots to make enough male nematodes to have an effect on the population. So we're working on solutions to that, but there's a lot of hundreds of millions of dollars of science gone into the malaria mosquitoes. We're just at the beginning of that, but I think it's a good place to invest, right? Because we know that the time we've got left with these resistant varieties, with the other things we're doing, is limited, and we're already starting to see problems, especially if you grow, if you try and grow continuous soy, or you don't rotate, you're going to see problems very quickly.
Good, thank you. So I guess you kind of covered a lot there. You shared about your research. I think one of the hot topics, or several questions that I've been asked throughout the season, and I've usually tried to talk with the researcher and get questions answered the best way we can. So if you have any questions on Soybean Cyst Nematode, we can work through those throughout the season. I can easily email, for example, you and get some quick questions or answers to the questions. So one of the questions or hot topics is definitely Peking. And for example, I know there's been farmers tell me that they've seen as much, or I read, as much as a 20 bushel yield bump with Peking. And do you guys want to comment on that? I know you gave a little background on Peking, but this is what some farmers are actually experiencing right now.
Yeah, so a lot of this is anecdotal. So the Peking bump is a big change from olden days when there was a Peking lag, where people didn't want to grow Peking because of this yield lag. And I think it does go to show what Matt was talking about, that the nematode is starting to adapt to the 88788 form of resistance. And so having that additional control strategy there, which really can knock down those nematodes that are adapted to 88788, is a good way to go. Again, the number of varieties out there for farmers, though, is limited.
How quickly can the nematode become adapted, or how quickly will Peking last?
I can take that one if you like, because we've done an experiment in another project on that one where we grew continuous soy in different plots. And we looked at Peking resistance, 88788, and a couple of newer exotic ones. And actually, 88788 is one of the most durable, but it's also one of the weakest forms of resistance. And then Peking's quite a bit stronger, but we did see a strong increase in virulence over a 6-year period. And then some of the newer ones, which are stronger, they get overcome more quickly. And what helps you sustain that is if you rotate the different resistance sources. So even if you're growing continuous soy, if you can go 88788 Peking, 88788 Peking, that's actually very durable in this experiment. So even if you're growing continuous soy, you can rotate your resistance, rotate your varieties, you'll get a better result. The other thing I'd like to say is I think the reason people are seeing this Peking yield bump is it's not really a yield bump. What it is the loss we're seeing that is slowly creeping in from these nematodes that are getting more and more virulent on the resistance source that we're all using. It's kind of like a zombie movie. They're slow moving, and there's more and more of them all the time, but you don't notice. They creep up on you, right?
Yeah.
And I think the problem is that the companies have done a great job boosting yield. And just, if you look at corn, people are used to getting bigger and bigger and bigger yields every year with the new varieties. They're actually doing that in soybean, but you're not seeing it because the nematodes are eating away that extra yield from these newer varieties. So when you put the newer varieties and you introgress those old resistance genes, it's not Peking, but it's a few genes from Peking like RHG4 and RHG1A, then you see this massive yield bump because suddenly the nematodes can't grow on that germplasm. So I do think there's, people are missing out on yield they could have. But yeah, as far as I know, you can't buy Enlist soybeans that are Peking type resistance or something like that. So most of them are public varieties, most of them are non-transgenic. So unless you're growing organic or something like that, there's a lot of reasons people want to grow Enlist, right?
Right. Oh, yeah. weed control, for sure. Next up, one of the other common questions that I get is having to do with nematode testing. And I know we've worked out a lot of kinks with that system as well. And I'm very grateful for you for working with us. And we talk about, certain things. I know there's people in this room actually that have done the nematode testing. So they may have some questions or comments too. But I appreciate you working with us to make the program better each year. And one of the common questions that I get is thresholds.
Yeah, and this is a question that I get a lot, and it's one that I kind of look down at my shoes at and mumble away something. So thresholds is something that nematologists don't feel comfortable with. And part of the reason is that it's a complicated system. When we take a soil sample, or you take a soil sample, you're taking it from one small area of the field. You could move over a foot, and you might get a very different result. So we're getting an idea of the populations there. That's one issue that causes uncertainty. The other area is that, and if you've taken plant pathology, Lauren, I know you took plant pathology, you remember the disease triangle. I'm sure you took it with me.
You'll be quizzed later, Lauren.
So the disease triangle has the pathogen, the host, and the environment. And so the environment plays a role in terms of, okay, how bad will a certain level of the pathogen be towards your host under that environment? So if you have great fertility, you're keeping weeds down, you get plentiful moisture, you may have a higher level of SCN and not see as much of a yield loss. At some point, a number of eggs, yes, we're pretty confident you're going to get yield loss. So if we start seeing numbers above 10,000 eggs per 100 cubic centimeters, I can feel pretty confident, even if you have a good crop, you're probably going to get some yield loss. But it's really, really variable. And so that's why we don't usually talk about strict thresholds. I mean, I kind of think of it akin to, you go to your doctors and you get your cholesterol tested, right? And you get a number. Well, what does that number mean? Well, it's going to depend on, okay, do you smoke? Did your parents have heart disease? Do you exercise? What kind of food do you eat? And all those things can go into it, and they can affect how you interpret that number. Do you have to go on a statin or not? Okay? It's the same sort of thing. We can give you a number. We've got a general idea that's bad or that's really good, and it's in those middle areas where we can give you an idea and we can help you interpret, but it's not black and white.
Okay, thank you. Lastly, I'll take it back to you. Why should ISA keep funding your project for SCN testing?
Yeah, I think that's a very reasonable question. So I think that, again, I'll use my cholesterol analogy. It's like, I cannot take a look at any of you and predict what your cholesterol levels are, right? You have to take the test to know it. You can't predict it. You might know, well, I ate a bunch of those hot dogs over there today, and so my cholesterol probably went up. But you need that number. Right? Without it, you don't know sort of what risk you're at for long-term health effects. The same thing is true. We can't look at a field and we can't say, looking at the soybean above ground, oh, you've got an SCN problem. We can say, oh, you've got kind of a weak stand. We can look at yield numbers, but then correlating that back to SCN without getting those egg counts is impossible. Right? And so really, I think the service is for those growers to be able to say, look, this is what I have. Whatever I'm doing is controlling it well. I got a 0, great. Well, okay, I got a 10,000. I need to sort of think maybe I'll do a couple of years of corn. I don't know if I'm allowed to say that in an ISA conference or not, but I think it's really for the growers to get an idea of how to interpret their fields and what they're seeing and what they should think about.
So one particular situation, and I just thought of another question for you before we go to the audience. My mind is going now. But let's say this has actually happened to me. I do a field visit. I'm the agronomist on the scene. I'm CSI. I'm trying to figure things out. I have a list of questions like a doctor. And one of my questions usually is always, have you done an SCN test lately? And the answer usually is yes. And this really happened. Great farmer. And he said, yes, I was one of the first ones that they found SCN in Illinois. And he was very proud of it when I've heard the story about the extension service, traveling, and I'm sure that was fun back then with the sieve and actually going to farmers on boots on the ground and finding SCN for the first time. He has not tested since then. What would you tell him?
Again, I would say you can't test once and then predict for all time what your levels are going to be at. Things change. I mean, like we were talking about, the nematodes adapt. Nematodes move around. Anything that can move soil can move these microscopic worms. And so if you tested back in the late 90s when the first SCN coalition came about, things have changed in 25, 30 years. So it could be that you're at a similar level, but again, you can't know just by looking at the above ground tissue.
Thank you.
Yeah.
So you shared with me something that I found very interesting, and I've been thinking about ever since you told me when we met, but you talked about, and you can go into more detail, how hard it is in breeding from the university standpoint. If you create something or you come up with something new, how easy is it for you to get that to industry, to get it to the farmer?
Yeah. And I will address that, but I just wanted to say, and this is one of the reasons here, so my lab does stuff in the lab, right? We're lab people. We've got nematodes growing in the lab. We've got soybeans growing in the lab. They live in these metal boxes. They don't go outside very often. Sometimes we'll do stuff in a field. If we do, it's that field over there behind the stands. That's the only field we ever do anything in. And a lot of the times you do this, it's not going to work on a farm, right? So there's a lot of this university research. It looks great on paper. It looks great in the lab. It doesn't work in the nematodes from the next county over, let alone the ones down in southern Illinois or near the river. And so if you don't have these projects that bring in nematodes from everywhere else, those allow us to check, at least in the lab, we can say, does it work on these nematodes? Does it work on these nematodes? Not just our little lab rat population. Mm-hmm. But to go back to what you were saying, I think the big limitation of a lot of this research is because of those things I mentioned, because so many academics do stuff in their lab and then they have a PhD student, that's their project, they graduate, right? And that's your aim, is you get your student, they write their thesis, they write a paper, and then they're done. Mm-hmm. And a lot of the industry folks, they look at that and they say, what's the chances of that actually being something we want to invest in? And when they take something from us, so it might cost us more than you think, so probably $200,000, $300,000 more to get a PhD student through the program and do that research. But then industry, they look at that, for them to take that product, get it to market, they're looking at $200, $300 million. And they've got to get it through regulatory, they've got to get it through FDA, they've got to get it through APHIS, and they've got to bulk this up, they've got to test it across the country and maybe also in other countries. So for them to take that bet, it has to be really, really compelling. And it's hard for us to persuade them to take it to the next level. And one of the things that's missing is a way for us to get better information to convince them, right? There's kind of a gap. We can't get those inventions even well enough characterized for them to get excited in most cases. Now, there are some honorable exceptions, especially with the breeders, and they have links, they have ways to do this. We have, Illinois Foundation Seeds and people like that, and so there is a way to do it for Jessica's oat varieties or Eliana's soybean varieties. But even then, it's like they get to be a public variety, but then getting that public variety then turned into something that is Roundup ready or Enlist or whatever, that's another huge investment, and only the companies can do that. And that's why we don't have Enlist Peking varieties. So I think part of the issue is the regulation is very expensive, and only a few companies can get stuff through it, particularly if it's a herbicide resistance trait or something like that. Mm-hmm. And part of the problem is just this disconnecting culture between academia, where it's partly about training students. And so if you're doing this to train students, it's probably not going to be super professional and super convincing to industry. So I think we both kind of have to work on that and build better links. And one of the things you can do is have your students go to industry and do internships and learn what their standards are for stuff, and that tends to help.
Okay, thank you. I'm going to open it up to questions and make Kelsey work. Are there any questions?
Here you go, Brandon.
I've got several questions here. So realistically, going back to taking the sample for the first time, you get a baseline. Realistically, how long should we wait before we're taking that next sample? You know, soil tests, we're waiting every 4 years. Is this something that we should probably be on the same rotation?
Yeah, so I'm glad you mentioned that. So I usually recommend doing it along with the soil testing. You know, every few years, you can set it to presidential election, whatever you want, but doing it on that cycle is pretty reasonable.
What's the wait time for getting those results back?
Yeah, and that was one of the issues that Stephanie was mentioning, the kinks in setting this up. So I'll give you the academic answer, which is it depends. It depends in part. We'll get obviously big push times where we'll get a crop consultant that sends us in 50 samples, and each one takes time to run. So we're trying to get the numbers back to growers within a couple of weeks, but around the holidays, a lot of the workers in the plant clinic are undergraduates. They're going home. So we're trying to get it within a couple of weeks, but there will be times when it goes over that. But
I just want to give kudos to the ISA and then your lab for that opportunity. It's a neat thing to be able to bring to my growers to give them an idea, especially these high productive growers that want to know, or get to that next level of yield to essentially eliminate or figure out if that's one of the yield robbers in our area. So thank you for that.
Great.
So I used to work at the University of Illinois Plant Clinic many moons ago, and at that time we had the same sort of program. So it was really nice to bring that back. And actually we had a lot of farmers that were asking for that. And so I think we still learn about what's going on out in the field, and I look forward to doing more types of surveys like that. And we have been funding some different types of surveys like that to learn more.
You can test at any time. I mean, the interesting thing about the test results is that that's also a factor in terms of how you interpret it. So if you're getting high numbers and you're going to go into corn, well, that's going to be a little bit less worrisome than if you have high numbers and you're going straight into soybeans. So what I tell people is any time you can get a probe into the ground. So I mean, the past few days, you might have been hard-pressed to go out there and really take some good samples. You can find SCN any time of the year. They don't go anywhere. They're just hunkering down over the winter.
Question right here.
As long as the ground's not frozen.
Yep.
How critical is the procedure from collecting them, getting them to the lab? I know it's supposed to keep it cool, refrigerated.
Yeah, it's actually a little bit less critical than with some of our other nematodes, like nematodes on corn, where we really need them to stay alive with SCN because we're only counting eggs. We don't want them to go too long because you'll start getting microbes in the soil that will start breaking down the eggs, and that can result in changes in the count. So we generally recommend keeping it cool, not frozen, but keeping it, let's say, 40 degrees or so, and getting it to us within a week or two, and that's fine. If we have samples that we have in our cold room that are months, probably even years old, that we can get viable SCN eggs from. So they're great survivors, which is one of the reasons they're so problematic.
Here you go.
Where are you with the OxyTech program? And I guess as a question, where are you with the Oxitec program? And then as a follow-up, is the hope that one day you would release the edited varieties such that they would kind of sterilize themselves?
Okay, so first of all, we're right at the beginning, and the Oxitec approach, that is patented by these guys. So, and that would be another complication. We're allowed to try it, see if we can get it to work, but if we did get it to work, we would need to work with them to be able to market it. But they might be actually a little more willing to look into this than maybe some of the big ag companies, because this is what they do, and they've done this in a lot of different organisms. But we're right at the beginning, and the limitation right now is you have to make males. So the first thing we have to solve is, how do you make male nematodes in large quantities where you can actually release them? And how do you release them? Can you do a seed treatment with male nematodes or something like that? And which genes are active in male nematodes? Because we need to put this gene in, and it needs to be active in the male specifically. So first thing we got to do is make lots of males, figure out what genes they got that are in the males, not the females. And that's what we're focused on right now. That, and actually I'm borrowing his microscope right now, because we need a special fancy microscope to be able to try and put the genes in. So the way you do it in nematodes, the standard way of doing it in other nematode species, is you get a very, very fine needle and you literally inject genes into the nematode.
But that's an expensive scope.
It's not the most expensive, but it's pricey. So, but yeah.
It's not like an electron microscope.
No.
Is that one of the most expensive?
Yeah, electron microscope will set you back $500,000.
Well, we've got one on North Campus that I think was $20 million.
Ooh.
Electron scope. So they get
We
up
don't
there.
have that much funding.
I've got a question right here.
In terms of managing for the nematodes, is there any validity to some of these biocontrols out there, either from like Indigo Ag with some, or like different chitosan products that have, just to try to regulate or control the feeding from the nematodes on soybean roots?
Yeah, so the question was about biological. Now, are you talking about seed treatments or more like
Or cover crops, or
any biological control. I believe, I guess, in the chemistry is somewhere we can at least set the populations back. But I've seen some, thoughts, I guess, on using a biological control like a bacteria that'll to where kind of a more season-long control than what the chemistry might be able to provide.
Yeah, so there's definitely there's a lot of research that's done and has been done, and there are commercial products out there, primarily seed coats, that are microbial. They're microbial seed coats. A few of them are supposed to coat the root and then serve as an antagonist, so detract the nematodes from attacking them. There's another one that is actually a microbe that will infect the nematode and kill it. A lot of the results that I've seen, I'll see some results where you get an increase in yield. I'll see some results where you knock down the nematode, but you don't see a yield increase. I haven't seen anything where it consistently increases yield and knocks back the nematode. So I think there's potential there, but I haven't seen something that I would bet the farm on.
I've talked to a couple of growers actually that had good results from that, but they said it worked the first year, and then after that, didn't do anything. So I think one of the problems is, just as with the resistant plants, these nematodes adapt really fast. And they probably adapt quicker to the microbes than they do to the plants, because plants have got more tricks up their sleeve.
Yeah, so.
Good question. Any others? So.
Do you have a question right here?
Oh, okay.
So in the next 5 to 8 years, what do you think the primary control for cyst will be?
Well, it's still going to be 88788. I mean, it's being used, and You know, I sometimes feel bad for being all doom and gloom about 88788, but the fact is it's still effective. It's still much better than planting a susceptible variety. You still get control from it. I mean, the fact that 20% of the samples that we got back are zeros, I think is testament to that it's very likely those growers are planting 88788. I think that we will start to, as more Peking varieties get developed and as other sources of resistance get developed. So I think it's still going to be resistance. I think, you know, some of the seed treatments companies are putting a lot of money into that. While I haven't seen anything that's really magic yet, you know, I'm an optimistic guy. I'd like to hope that that's going to happen. And yeah, so, but I think resistance is the mainstay.
I think it's always going to be resistance. We just need to help the plants out any way we can because they're under attack, right? And I think that, but if that yield bump, if you're really getting 20 bushels from Peking and people are seeing that consistently, the companies are, that's going to move the needle, right? And they're going to start putting Peking type into their elite Enlist lines and stuff like that.
We just need Eliana to work faster, you know, breeding plants. Yeah.
No pressure.
And there's a lot of new resistance sources in the pipe after Peking too. So actually we have a good lineup to go to. It's just getting them into those farm-ready, high-yielding varieties.
Any other questions? So I have, if Kelly Robertson were still here, he'd remind me that there's a lot of customers, especially his customers, especially farmers down in southern Illinois, and not just there, that are planting soybeans on soybeans. And they're still getting great yields. I hear that a lot. What would your advice to them be in regards to Soybean Cyst Nematode?
Well, I would say, you know, be careful. Keep that testing up. You know, again, it could be, if it's effective, great. And if you're getting really low SCN counts, that's fine. I think the issue is that SCN, like a lot of plant parasitic nematodes, is really sort of insidious in that you don't see obvious above ground symptoms. And so if you're happy with your yields, that's good, but would you be happier with higher yields? Probably. And so I think, you know, that's the question, is what would you be getting if you had effective SCN control?
It's the zombie movie again, right? There's always the guy that's like, oh, I don't believe in zombies. And you know, he's always the first one they come for, right? Yeah. But there are populations. There's a lot of variation in these populations, and we see that. And there's probably some local populations that find it harder than others to adapt to that 88788 type resistance, just through luck. But things move around, right? They move around on the wheels of tractors and harvesters, and you know, you don't know when you're going to get the bad nematodes in your field if you've got the ones that the resistance is working on right now. So as those populations get mixed up and expand, you might not have a problem now, but you might in the future. So again, I think keeping up with your counts, because there may be, you know, the zombies may be starting on you and you haven't seen them yet.
What do you say to those guys in the South?
Ooh, I'm coming.
There you go. Wouldn't you say the guys in the South, you're talking about the South, but they don't know what they don't know. So that's why we have to do this testing to even give us a baseline of what, you know, what's going on. So they probably don't even know that they have a problem or they don't have a problem. So yield really, so they feel like the yield is really good, but they could, like you said, could be more, but they just don't know what they don't know.
And they don't have a check, really.
Right.
I don't have a question.
Okay.
Very good.
All right. All right, anything else before we wrap up here? Any last things that you would like to talk about? I'll give that opportunity, and then I have one last question. Anything that we didn't address?
Just to reiterate, free SCN tests. So free SCN testing at illinois.edu. you can email us and find out more information about where to send samples, how to send samples, all that good stuff. Yeah, if you have any questions, feel free to reach out.
And yeah, well, and thank you for all the support and input, and it's really useful to know what it is people are needing. It's also useful, I think, to understand that despite what we hear elsewhere, there's a lot of people having trouble with SCN out there. And just knowing that there are farmers that are struggling with it and that they want to continue to support us, that's really important. And we're happy to respond to any and all comments and questions.
So I think Nate pointed out to me, and I feel this way too, from a plant pathologist / I didn't go into nematology, there was a reason. And if you've ever done SCN testing, it is not fun, just so you know. It's about as fun as being a soybean breeder. It's not one of your top jobs. Would you say it's probably, it would be hard to find a worker, probably, to be completely honest, to do that type of work.
I actually kind of like, I love looking under the microscope.
We're happy you like it.
Yeah, I like it. I wish I had more time to.
Yeah, so I went more in the disease side of it. But with nematodes, we talk about it all the time. And I know, Pam, you've written about this constantly. And I think we almost get to a point that we become numb to it. And so what would you both say to that? And what is your message to farmers?
Don't become numb. I don't have a great strategy except to say, it is out there. It hasn't gone away. It's not going away anytime soon. Yeah, it may have lost some of its sex appeal compared to red crown rot or tar spot or any of those other diseases that are really in your face. But it's there. We still consider it to be the most important yield-robbing pathogen of soybeans in the Midwest. So, yeah, keep an eye on it.
And yeah, I mean, if you're growing the same lines from the same supplier year after year after year, and you may think that they're great, but maybe it's worth trying a small area of another variety if you can get hold of it, just to see if that does give you that yield bump, because you may just be benefiting from all this genetics, all these billions of dollars the companies are spending inching that up, and it's keeping you flat. But actually, the nematodes are robbing you of what you could have there.
So to end, if you had a wish list for the ISA, Illinois Soybean Association, what would you ask for? If we were Santa, what would you want? How can we help?
Well, I've got something, because
I'm catching him off guard, by the way.
I think ISA, USB, North Central, you guys have the ears of a lot of these companies, and because you're farmer-driven and farmer-directed. And I think you can really help us kind of spread these messages. Like sometimes we say these things, and sometimes the farmer's like, I'm fine, maybe a few people have this problem, but if you're all feeling the problem, then if you start asking for Enlist, approved Peking type resistance, then if you start asking the companies for it, they might do something about it. They tend not to listen.
And I think we have a similar issue if Dr. Bond was here with root knot nematode in the South. And so we're trying to get the word out. Root knot nematode is there. And we have a lot of varieties that don't have, in our maturities that we grow down south, that don't have the root knot nematode resistance in them. So that's another way to add to your comment, another example of that story.
Yeah, I guess what came to my mind, I was just asked to contribute to this book chapter on lesion nematodes. So this is a different genus of plant parasitic nematodes, Pratolynchus. They're everywhere in Illinois. They oftentimes get referred to as corn nematodes because they do feed on corn, but they are also happy feeding on soybeans. They don't care. As long as they have a plant to feed on, they'll feed on it. We have no idea what their impact is on soybean. They're all over the state. When we've done corn surveys, we find them in 80% of fields. There are other plant parasitic species. How are they interacting with Soybean Cyst Nematode. They may be, contributing sort of a synergistic effect, and we don't know. So I think there's a lot of open space in that area to think about, okay, what are these other organisms doing?
Okay. Well, thank you both so much for being here.